Hi all, today we are discussing about types of restriction enzyme. Introduction. During 1970s, it was found that bacteria contain nuclease that would recognize short nucleotide sequence with duplex DNA and cleave the backbone at highly specific sites on both strands of the duplex. These enzymes are called restriction endonuclease or simply restriction enzymes. They were given this name because they are used by bacterial to destroy various viral DNAs that might enter the cell, thereby restricting the potential growth of viruses. These are of different types, and the types are almost explaining here. Contents are restriction enzyme, type 1 restriction enzyme, type 2, type 3, type 4, and reference. Restriction enzyme. A restriction enzyme is a protein isolated from bacteria that cleaves DNA sequence at sequence specific sites, producing DNA fragments with a known sequence at each end. The use of restriction enzyme is critical to certain laboratory methods, including recombinant DNA technology and genetic engineering. Type 1 restriction enzyme, capable of both restriction and modification activities, contains separate restriction, methylation, and DNA sequence recognition subunits. They cleave DNA far from recognition sequence. Example, ECO-K1. Type 2 restriction enzyme. These are most commonly available and used restriction enzyme, composed of only one subunit. Recognition sites are usually undivided and palindromic and 4 to 8 nucleotide in length. Recognize and cleave at the same site. Do not use ATP for activity. They are exclusively used for DNA analysis, gene cloning in the laboratories. Discrete restriction fragments and gel banding patterns are observed. They are named after the bacterial species from which they are isolated. Thus, its example is ECO-R1 and it is isolated from E. coli. Type 3 restriction enzyme. They recognize two separate non-palindromic sequences that are inversely oriented. They cut DNA about 20 to 30 base pair after recognition site. Contain more than one subunit. Require a adomate and ATP cofactor for their role in DNA methylation and restriction. Example, ECO-P1. Type 4 restriction enzyme, methylation dependent, cleave only normal and modified DNA. Recognition sequence has not been well defined. Cleavage takes place approximately 30 base pair away from one of the site. Its example are not identified. This is a table regarding the cleavage site, allocation of methylase and examples of type 1, 2 and 3 restriction enzyme. And thank you.